Well, first of all, I'd like to thank Pat and everybody at Parent Product for inviting us to come here today and to talk with you both individually and to also share our data with you. It is incredibly meaningful for us to spend some time with the Duchenne community and to understand, to talk with you one-on-one, and I think one of the previous speakers talked about it being very energizing. And it's very energizing for our team to go back and really try to do the best we can for moving things forward for the Duchenne community. So this afternoon, I'm going to talk with you about Edasalonexent, where we are in the development. We're in the middle of a phase 2 program, and we have a number of results and are encouraged in moving forward. So I'm going to share that with you. We are a public company, so I do need to share a forward-looking statement, and of course my conflict is that I work for Catabasis. So Edasalonexent is an oral molecule that inhibits activated NF-kappa B, which is a protein target that itself is activated very early in Duchenne, and it has a number of effects that cause muscle degeneration and slow muscle regeneration. So our aim is, by inhibiting NF-kappa B, to slow muscle degeneration and allow muscle regeneration. Now, NF-kappa B is activated by mechanical stress in muscles, and it's actually most activated and causes inflammation in the large muscles of the leg first, because they're under the most mechanical stress. So with activation of NF-kappa B, there are a number of downstream effects, and there is inflammation that is mediated in large part by NF-kappa B that then goes on to trigger fibrosis. It also suppresses muscle regeneration, and when muscle is being destroyed, of course, it's even more important to allow it to be regenerated as quickly as possible. And it specifically, an activation of NF-kappa B degrades proteins in muscle, so there's a number of effects. Now, steroids actually target NF-kappa B, but they are very good at decreasing inflammation, but they also themselves have effects that suppress muscle regeneration and also drive muscle degeneration. So we're hopeful that by targeting NF-kappa B, we can have the good effects of steroids and perhaps more, but importantly, not have the side effects that you just heard it talked about, the osteoporosis, the fracture risk. And our hope is to have a better therapy to use in boys and young men, regardless of mutation type and across all ages. So we have shown in a number of animal models of Duchenne that inhibiting NF-kappa B with Edasalonexent has these positive effects. Now, another preclinical study that we've done before I turn to the clinical studies is the hope is that these therapies can be used together. And inhibiting NF-kappa, with activation rather of NF-kappa B, one of the things that it does is it actually suppresses dystrophin production. So in states where there is some dystrophin, with exon skipping, with Becker's, the activation of NF-kappa B and the inflammation suppress dystrophin formation. So we've been working with Sarepta to look at what happens in the combination of the Eteplirsen analog in the mouse with Edasalonexent. So on the left panel here is dystrophin or rather the lack of dystrophin in the MDX mice. And with 4 weeks of the exon skip that's analogous to Eteplirsen, you see that there is some dystrophin formed already in 4 weeks. Then with co-administration of the exon skipping therapy and Edasalonexent, you see even more dystrophin. So by a separate assay looking at the amount of protein here, and it doesn't show up, it goes from 4% to 7%, so almost doubled. So we're hopeful that ultimately the combination will also be helpful. And to that end, what we are doing in the current and future clinical trials will be to allow boys that are eligible for eteplirsen to start on that. So we're starting combination therapy with an investigational drug, Edasalonexent, as well as with now a newly approved drug, eteplirsen. So the study that we've been working on the last year and a half, and we're enormously grateful to those of you in the room who've been working on it and your sons participating in it, because we know it is an enormous burden. The MOVE-DMD trial was a two-part trial. We started out after phase 1 studies in adults, and we started in a phase 1 part with boys with Duchenne, boys aged 4 to 7 that were not yet on steroids. So we started out with 7 days of dosing. We showed that that was safe, well tolerated, and the exposure was similar to what we had seen in adults, where NF-kappa B was suppressed. We took baseline measures of function and also MRI at that time, and then conducting the part A of the study and getting ready for the next part of the study, the phase 2 study, took on average about 8 months. So when the boys started in a 12-week placebo-controlled study, the part B, we had some information on how their functional measures changed over that 8-month period, so we could compare that. And part of the design of the study was not only to look at the placebo-controlled part of the study, but also to compare how things changed when the boys were off treatment with how things changed when 12 boys crossed over and started on Edasalonexent. And that's actually a more powerful statistical design incorporated into the study. Right now we're in the open label extension part of the study. Boys have been now dosed for over a year with Edasalonexent, and we're moving ahead with that. So the study was conducted in conjunction with Imaging DMD, conducted at their sites, as you see, typically on both. We missed flyover country in the middle, clearly. And we looked at MRI as an early biomarker. This was the first time it was used as an initial and exploratory endpoint, as a primary endpoint in the study. And we also looked at functional measures that are appropriate to boys of this age, with 10-meter walk-run, 4-stair climb, and time to stand from supine, but not 6-minute walk. And we also looked at North Star and a pediatric, the PODC, the Outcomes Data Collection Instrument. We have, basically this shows the overall results of the study. And what we saw in the 12 weeks of the study, the pre-specified analyses, it looked at 2 doses of Edasalonexent. The high dose, the 100 milligrams per kilogram dose, we saw that Edasalonexent, the effects were positive compared to placebo in all 5 functional tests, the 3-time functional tests, the North Star, and the PODC. And at the lower dose, the 67 milligrams per kilogram per dose, the Edasalonexent treated boys did better than the placebo boys on average in 4 of the 5 measures. We also looked at what happened to the boys that were in the initial part of the study and what happened when they crossed over onto Edasalonexent. And again, all 5 of the measures were favorable. So we did not see statistically significant changes in the MRI, although they did move in the right direction. But what we really wanted and what's important is the functional measures, because that allows us to design a phase 3 study, and of course that's what's going to be used for registration and ultimately for approval of these drugs. So we also, we're very encouraged by this. We also did a composite measure to look at the overall effects. And in this analysis gave basically 1 point for each of the functional measures. Did they get better? Did they get worse? So +5 means they got better in all 5, -5, they got worse in all 5. And the boys who were taking placebo for 12 weeks, on average, got worse. And we had seen this in the off-treatment period as well. So they generally did not improve. And the boys who were on Edasalonexent generally were either stayed the same or on average even improved a bit. And this difference was statistically significant on this global outcome measure. This is a study that is with 31 boys overall, and it's not powered for these functional measures. This is a study in order to be able to then design a phase 3 registration study. So we're very encouraged by this. I'll just walk you through a couple of the time function tests. We have more on our poster, which will be available as well electronically. And with the 10-meter walk-run, what we saw with the black bars, the vertical axis is the change in speed over a 12-week period. And we saw that both during the placebo-controlled phase on the left, as well as during the crossover phase, the off-treatment period, the boys got worse, unfortunately. With Edasalonexent, there was a decrease in the rate of decline. So in the crossover group, it was 50% less. And these boys, if you can think about if you can keep them from getting worse, then and extend the time to ambulation, the time to keep function, then that's very important, particularly without the side effects of steroids. We also, one of them that we looked at was the pediatric outcomes data collection instrument, and that we asked the parents to comment on and answer questions about what's happening over the last week. Are the boys able to do with ease, with difficulty, a number of measures, walking a block, climbing stairs, getting into bed, functions that correlate, that Craig McDonald has shown correlate very well, this type of questionnaire, with functional measures and ultimately and loss of functional measures. And there we saw that in the placebo period, the off treatment, the boys were getting worse, and on Edasalonexent, they were getting better. And in fact, in the crossover study, that was statistically significant, even in a very small number of boys. So safety is obviously key. We did not see safety signals. The most common adverse event is mild diarrhea, and which tended to be transient at the beginning of the study. There were no serious treatment-related adverse events, no patients who had to reduce the dose or discontinue because of a safety issue. So what we are concluding from the phase 2 study is that we have seen that in contrast to the off-treatment period, to the placebo period with these boys, we're seeing a reduction in the rate of decline in functions in this study. We've seen this in the well-established functional tests that would be a primary endpoint in a phase 3 study. And we've seen improvements actually in scores on validated global measures. And in some ways, maybe it's more meaningful to look at what parents see has been happening over the last week rather than the few seconds doing a time function test. So we're encouraged by those observations. The safety has been excellent to date, and in fact, we've moved all of the boys to the higher dose, the 100 milligrams per kilogram dose, because it was so well tolerated. And this gives us the information that we need to be able to design a phase 3 study, which is what we're working on at this point. We have more information coming in from the open label extension, and we'll be analyzing that and sharing that in the next several months in the third quarter. And we expect that we'll be then announcing what we're going to do for a phase 3 study in this young population of boys not yet on steroids sometime later this year, and anticipate then potentially starting the study soon in the next year. So, and lastly, I would like to thank everybody who has contributed to the completion or to the study and all of the information we have. First and foremost, the parents and boys who have been traveling crisscrossing the country, there is and undergoing the testing, which is it's a considerable burden. Obviously, we know that in the phase 2 study. The advocacy groups, the patient groups that have helped us, and in particular PPMD and MDA have provided travel funds for supporting the families in this study, and the team in Imaging DMD. So thank you.